Great. Okay, well, I'll make a start. The next presentation for this afternoon is going to be on the Tuam Mothers and Babies Home possible options for DNA testing. Now, there's been huge advances in DNA in recent years, even in recent weeks. And the big question is, how can these rapid advances in DNA technology actually make it easier for us to solve some of the problems and right some of the wrongs in relation to the Tuam Mother and Babies Home. Now, for those of you that don't know a huge amount about it, I will be talking about the advances we made when we identified the remains of Richard III. I'll also be talking about the project at Fromell, where 250 World War I soldiers from Australia were found in a mass grave in 2009. And through the use of a combination of DNA and other artifactual evidence, so far we've managed to identify 159 out of the 250 soldiers. So the methods that were used there could potentially be used also in Tuam. I'll also be talking about some recent advances we've made through the work of teams in Ireland, such as Professor Dan Bradley's team at Trinity College Dublin, which analysed the DNA profiles of skeletons that were found under a pub on Rathlin Island off the coast of Antrim, and these skeletons date somewhere between 4,000 and 6,000 years ago, and they have changed the way that we think about Irish genealogy and Irish history. So Professor Dan Bradley's team used whole genome sequencing, so they got the entire genome of these skeletons, and that caused a minor revolution, certainly in the field of ancient DNA. And we'll also be talking about the identification of Thomas Kent, which was carried out by the Ancient DNA Lab at UCD, University College Dublin. Jens Carlson was the person that spearheaded this particular project, and his presentation can be viewed in full on the Genetic Genealogy Ireland YouTube channel, and that's the link there. So we're talking a little bit about all of these particular projects because they have helped advance the study of ancient DNA, and also they have applications for genetic genealogy and specifically for the identification of the children at the Tuam site. Now, for those of you that don't know a huge amount about it, and I know that some of you have come from afar, so you may not be familiar with some of the background details relating to Tuam, but there was a mass grave known to be there from the 1970s when children playing on the site discovered that there was a slab, and they looked through and they saw lots of skeletons of children in this mass pit. Then in 2014, Catherine Corliss, a local historian, published the fact that there were 796 children who died at the children's home in Tuam, and there was no record of where they had been buried. So the question was then, were they buried in that mass grave that had been discovered underneath the memorial garden that the locals subsequently built to commemorate these children? These children died between 1925 and 1961. Here's a photograph of the actual memorial garden, and the mass grave is beneath the grass here. It's beneath the grass, and then of course it hit the headlines, so we have headlines like remains of children found in sewage chambers, that then Taoiseach Enda Kenny said we took their babies and gifted them, sold them, trafficked them and starved them. The next steps in finding the truth behind their deaths, so people want to find out exactly why these children died. It's important we get this inquiry right. The mixing of the remains will make it very difficult to identify babies at Tuam, and then a group recently from Trinity and UCD have said that no, with modern DNA techniques it is possible to have identifications at Tuam. So we're going to look at some of the issues around the Tuam site and to see how DNA can possibly help answer some of the questions that we have. The names of all 796 children have been published. They're public knowledge, and there you see a list of them on Irish Central, but they've been posted on other websites as well. I took the first of those names, Patrick Derain, and you can see that his death record states that he died on the 27th of August 1928, He was 5 months old when he died. He was the son of a farmer. He died of gastroenteritis, which he had for 2 months. And the informant was Sister Hortense, who was the occupier at the children's home in Tuam. And many of the children in the Tuam children's home were illegitimate. There was a huge social stigma associated with illegitimacy back in Ireland in those days, and that was still there up until the 1960s, into the 1970s. But because we have the names of these children, it would be possible to generate family trees for each of these children, especially if they had rather rare surnames like Durain. But of course, because they were illegitimate, we'll only be able to research the mother's side of the family tree. We don't know actually who the father's side is. The Tuam Home Survivors Network published a press release in January of this year, and they said in relation to the children at Tuam, there were 3 areas where DNA could possibly help. So if you look at the first area, that's number 3, the recovery and relocation to be undertaken with all the expertise and resources necessary to preserve, as far as possible, the individual identity of each set of remains. And that's a big problem because over the passage of time, the body decays, the skeleton emerges, and then of course the bones get separated. So that's one of the problems, is trying to put the pieces back together. And DNA can help in that regard. The second one is point number 5, the taking and cataloguing of the DNA from all remains to create the most complete database possible. So this is creating a database of the children in the mass grave. And then the third one where DNA could potentially help is that they want to extend an invitation to all those who have reasonable grounds to believe that members of their family may be buried at the Tuam site. And they'll be invited to provide their DNA so that their DNA can be compared against the DNA of the database of children at the Tuam site. So those are the 3 particular areas where DNA could help. And so from the point of view of the Tuam Home Survivors Network, the current objectives are really to try and preserve the individual identity of each child, to create a database of all the children's DNA, and then to invite selected families to provide DNA for comparison. And if a child is identified, then the remains of that child can be released to the family, and the family have the choice of burying it in the family plot, perhaps with the mother of the child. So that's the kind of thinking currently. Then there was an expert technical report produced at the request of the government and published in October of last year. And this is a very large report. There's 219 pages. The main body is about 80 pages, and then the rest are appendices from various groups who did the geophysical survey and so on. It's an excellent read. There's some wonderful recommendations in this report. There's lots of great examples of previous mass graves and how forensic genetics has helped identify the people and reunite the families with the people that are found in the mass grave. This type of action is described as humanitarian forensic action, and is carried out by forensic geneticists and forensic scientists who are more usually involved in finding killers, finding criminals, that type of thing, but where there's a mass grave, for example, because of an airplane crash, where there's not necessarily been a crime committed per se, they describe it as humanitarian action when the main focus is really to identify the people that have been involved in this particular accident or where a mass grave is discovered. Now, they say there are multiple challenges to be faced at Tuam's site. They reiterate on many occasions during the report that a pilot study would have been preferable before we produced our report, but the government wanted the report at a particular point in time, and so there wasn't time to do a pilot study. Now, that was last October. I don't know if they've done a pilot study in the meantime, but if they have, it certainly would be very helpful if a supplement to the original report was published with the results of that pilot study available for the general public to digest. So if there has been a pilot study in the meantime, it certainly would be nice to see what they found. One of the main suggestions that they put forward in the report was the formation of a multidisciplinary body to oversee and implement the strategy for dealing with the situation at Tuam. And I think that's a wonderful idea. We do need to have a lot of different experts with a lot of expertise, from lots of different fields coming in to advise on what's the best way forward. They also mentioned on several occasions a transitional justice process, like a Truth and Reconciliation Committee, that type of thing, which frequently happens when you come across these mass graves. They also make the point that forensics as a science is not standardized. And it points to the fact that different people do different practices in different countries, but also that the science is evolving and changing all the time. And when you look at the examples they give in the report, it's almost like every time there's a new mass grave found, the science advances. They learn something new from dealing with every single mass grave. And Tuam is going to be no different. And I think in many ways that would be a wonderful legacy for the children of Tuam if the exploration of the site actually advances the science of forensic genetics and forensic science itself. So that's another interesting aspect of this case. And they make the point that there have been many recent advances, and particularly with the DNA aspect of things, those advances have been extremely rapid. One also gets the feeling that many of the recommendations and suggestions in the expert technical report by the group have been ignored so far. And I think it's a very good report, and I would like to see the government take action on a lot of the suggestions and recommendations in the report. Now, here's the geophysical survey that's happening in the above the Memorial Garden, and they're looking into the chambers down below that patch of grass. This is what it looks like from the aerial position. You can see that there's the statue there over in the corner, and this is the area of the Memorial Garden, and the actual pit where the bones lie are beneath the surface. And this is the Memorial Garden here. It's a Google map with superimposed an old map of where the Tuam Babies Home buildings used to be. So you can see that the buildings used to be this area here, extending over here up to the top of the picture. So that is where the pit lies. And in the appendices to the report, you can see some very nice diagrams of how the pit occupies approximately about 50% of the area of the Memorial Garden. And there are various suggestions of how that can be excavated, and there's about 4 or 5 different options that they're suggesting in the appendices to the report on how this place can be excavated. But what we're looking at is an underground chamber, and there's about 20 of these. The whole pit consists of 20 underground chambers. There's bones in about 17 of the 20 chambers. So 3 of the chambers don't contain any bones. It has been entered previously, and talking to Catherine Corliss, there are piles of bones, and children have fallen into the pits, and that, of course, creates the risk of contamination, because the children will be giving off DNA. Every time we breathe, we put DNA out. So one of the things the team will have to do is take not blood samples, but DNA samples from people who perhaps have been in the pit, so that we don't confuse their DNA with the children's DNA. Another aspect is that apparently people who've been in the pit, and I'm not sure how you get into the pit, I don't know where the entrance point is, but they've seen bundles would have been left in kind of shelves on the side. So how they were placed there is another question. And of course the person that placed them there, their DNA will also be in the pit as well. So we have to try and distinguish what is DNA that is contaminating the scene, and what is DNA that's actually from the child themselves, from the children themselves. This is what the excavation scene looked like at Fromell. So this is where the World War I site, so it's about 90 years old, and you can see that there is an inflatable canopy over the entire site. People are wearing these white boiler suits to prevent contamination, and it is a crime scene investigation-like procedure. In Tuam, if they are going to be working underground, they may have to wear whole body suits, like a deep sea diver. And you can only stay in those suits for 40 minutes, and then you have to come up, take them off, have a breath, then a couple of hours later put them back on again and go back down, but just for another 40 minutes. So it will be a painstaking procedure, but it is possible. It's possible to do it. Everything they find down there will be logged. So they take a log of absolutely everything, put a chain of custody together, and of course at the Fromell site, where you're looking at 250 World War I soldiers, you're also looking at belt buckles, you're looking at maybe the remains of an overcoat, you're looking at maybe an insignia that was on the lapel. So there will be artifactual evidence around these soldiers that we won't have at Tuam. At Tuam it'll just be the bare bones, I think, and that's about it. The possible recovery team that we may have at Tuam will include a forensic archaeologist, an anthropologist and osteologist who may determine the height of the child, whether it was male or female, the approximate age of the child, and any distinguishing features such as bone fractures, diseases of the bone, which may even point to the cause of death as well. There will also probably be an historian involved, a photographer to photograph everything on the scene, and a project manager to keep things moving effectively. And the genetic team will possibly consist of a forensic geneticist, maybe with or without a genetic genealogist to advise, and there may be a statistician involved as well, because with the interpretation of the DNA results, statistics plays a very, very important part, and we'll take a little look at that. And we need somebody with an experience of doing this type of activity, somebody who knows about the ethics of digging up a mass grave and the potential sensitivities around revealing the identity of the children at Tuam. This is a diagram from the Fromell site, and the World War I soldiers were laid out in relatively neat graves. The Germans who laid out the dead bodies were very respectful. They even put brothers lying side by side. And that is the type of diagram that will first of all be done by the forensic anthropologists. They'll record the position of the remains. Every individual bone is mapped on these diagrams. Some will be intact, some of the bones will be scattered. The challenge will be to recover intact individual remains and rebuild individuals from scattered bones. DNA may help, but it may be too destructive. So for example, in Richard III's case, they used a molar tooth to extract some of the DNA, but in order to extract the DNA, they take the tooth, they clean it, they freeze it, they pulverize it and turn it into powder, and then they put the powder through various solutions, extract the DNA from that, but of course, at the end of that, you don't have a tooth left. So when Richard III was being buried, the scientists involved had to take the test tubes that the tooth had been in, scrape the inside out, and make sure that every possible remains of Richard III was put into the lead coffin, and then that was buried in Leicester Cathedral. So that's one of the problems, is that the DNA testing can be destructive of the smaller bones, not the larger ones, but some of the smaller ones. And the question is there, of course, what do we do with that? And there's a very good YouTube video about the forensic science group that did this, and it's available on YouTube, and you can see it there. This is the Fromell site, and you'll see in the distance that is where they found the mass grave, and they have those two inflatable canopies. And then they actually built a temporary mortuary and a temporary laboratory just beside the mass grave. And it was the samples for DNA testing were extracted at the site and then shipped from France all the way over to London, and in London then they extracted the DNA. So it is quite a complicated process. If it were to happen in Tuam, and here we have the Memorial Garden there in the middle of the picture, the question is, where would we actually build a temporary mortuary and a temporary laboratory? Is it going to be just south of the cemetery? Or is it going to be in this field here? I don't know who owns it. But these are the kind of practical, technical, logistical questions that the team will have to address. So how can DNA help? Well, it can help in three different ways. First of all, the identification of individuals, which is the obvious one, but also the reunification of bones, putting bones back together by testing each of the bones for what DNA do they have, and then putting the person that owns the bones, putting them all back together again, and also the discovery of trafficked children. Now, Enda Kenny, then Taoiseach, talked about trafficking children. Peter Mulryan, one of the members of the Tuam Home Survivors Network, he discovered through Catherine Corliss's work that he had a sister who was reported to have died in the children's home. But the question that arose in some people's mind was, has my sister really died, or did they forge the death certificate and she was actually sold to an American couple for adoption? So one of the things that we can do, if you think that your relative was trafficked, is do the Ancestry DNA test. Then when you get the results, download a copy of the results to your computer and upload those results to Family Tree DNA, MyHeritage, Living DNA, and GEDmatch. So that way you're swimming in 5 data pools instead of 1. And you can also test in the last of the major companies, 23andMe, and that will give you access to a database of 15 million people in total. So, and this is exactly the same advice we give to adoptees. You want to find out who your mother was, you want to find out who your father was, this is the technique that you would use so that you're swimming in as many data pools as possible, and hopefully in one of those pools you will catch the big fish. And it is getting easier and easier now for adoptees to find their birth parents using this method, and in about 2% of cases their parent is actually waiting for them in the database when they join. So it's an instant reunification, reconnection. So that's the first thing, child trafficking. The second issue is individualization, and I'm not clear at this stage what DNA tests would be best to actually do this. Do they use the standard forensic ones, or they do some of the more modern ones with ancient DNA? So these are still questions that would need to be discussed. Smaller bones, like I said, may be destroyed by testing them. Where do we draw the line? And we need expert specialist advice in this regard. But that is one other aspect where DNA can potentially help. But the one that I want to talk about for the rest of the presentation is identification. And the process of identification involves several distinct steps. So first of all, you have to get your tissue sample. Now, for those of us here doing genealogy, we'll either give a cheek swab, or we'll do the spit test with Ancestry, and we'll give some saliva. And the cheek swab will dislodge some cells from the inside of our cheeks. The saliva will contain white blood cells, among other cells, and they all contain DNA, and that is how we get a sample for the DNA work that we do as genealogists. If you're going to your doctor and he's doing a genetics test to test for some sort of medical risk, he might take a blood test. But if it is a deceased person, you're really looking at bone samples or teeth samples. Those would be the ones that are generally used, and the bone sample that we would tend to use is the petrous part of the temporal bone. It's called petrous because it's like rock. It is the most dense bone in the human body, and Professor Dan Bradley's team at Trinity College Dublin has shown that the yield of human DNA from the petrous bone is much better than from other bones. From other bones, because they're in the soil, so you're going to get contamination with bacteria and bacterial DNA. If you sample, say, a rib, maybe 10% of the DNA will turn out to be human DNA, and 90% will be bacterial DNA from the soil. But with the petrous bone, they've managed to get up to 58% human DNA, and the rest is bacterial DNA. So people are very much shifting in favor of the petrous bone to get their ancient DNA samples. In terms of DNA extraction, there's two aspects to think about: quantity and quality. How much DNA are you able to get out from a sample? And I know some of you probably have done a DNA test and you've heard from the lab about 2 weeks later, we got your sample, but there wasn't enough DNA in it. You know, you need to swab for 60 seconds, not just 5 seconds, or you didn't give us enough spit in your sample, please can you do it again? So sometimes there isn't enough DNA in the sample to do any testing of that DNA. And the other big issue, of course, is one of quality. Now, all of us are alive, so our DNA is going to be intact. But as soon as we die, our DNA starts degrading and fragmenting. And the older it is and the more it degrades, we go into smaller and smaller fragments. And it's much more difficult to test fragmented DNA than it is to test intact DNA. In terms of the actual testing that's done, we saw this morning 3 types of DNA: Y DNA, father, father, father line; mitochondrial DNA, mother, mother, mother line; and autosomal DNA, all of your ancestral lines. There's 2 types of DNA marker, the STRs and the SNPs, and I'll talk a bit about them in a second. And there's 3 approaches. You can do the forensic approach, which is what they do at crime scenes. You can do what I call as the commercial approach. That's done by commercial companies like the ones we use for our genealogy, Ancestry, 23andMe, so on. And then there's the ancient DNA approach, and that is the approach that people like Professor Bradley and Jens Carlson have been doing with the work that they do at their labs with the ancient DNA from the Rathlin Island skeletons, and also Thomas Kent. The work that was done on Thomas Kent created, again, new methods that will inform the investigation at Tuam. And then as soon as you have DNA from anyone, it's no good unless you can compare it to something. And for that you need to have comparison, DNA for comparison, a reference data set. And that can either be an ante-mortem sample, so a sample from the same person that was taken before their death, and we'll talk a little bit about that. It can be targeted individuals, like they did at Fermel, or it could be the general population, like, for example, the GEDmatch, for example, is an example of a general population database. And then to establish the identity, that's where the statistics come in. It's never absolute. You'll never get 100% definite. It'll be 99 point something. And, but what threshold should we use for the children of Tuam? Is it 95% enough? Or does it need to be 99%? So those are the kind of questions that the multidisciplinary body that should be set up to explore this need to address. So let's first of all look at the Petra's bone, and this is a picture of Kendra Sirac using the new sampling method, and Kendra is at University College Dublin, and this is a method that she developed that is now being used around the world. And it is a very useful illustration of how Ireland actually has developed the methodology that people around the world are using. In terms of the quantity and quality, well, DNA fragments after death, and here it is fragmenting. You can see that there's the double helix there, and then you have all the bases, but now it's fragmented, so you don't have it in one long strand, and that creates problems for some of the forensic tests. The fragmentation is not so much dependent on the age of the sample as the environmental conditions in which the sample is placed. So, for example, Neanderthals. They found some bones from Neanderthals that date back to 38,000 years ago, and they extracted DNA from a femur, thigh bone, back in 2010, and they were able to sequence the entire genetic profile of our ancient cousins. So much so that they found that we shared 99.7% of our DNA with Neanderthals. We share 98.5% of our DNA with chimpanzees, and we share about 99.8% of our DNA with every other human on the planet. So we are very closely related to the Neanderthals, and we've interbred with them, so much so that everybody in this room shares roughly between 1% and 4% of their DNA is Neanderthal DNA. On the other hand, if you look back just 100 years ago to the Titanic, which sank in 1912, 121 of the victims were buried in Nova Scotia, 42 were unidentified. In 2001, they dug up 3 of the graves, and they found that in 2 of the graves, none of the remains were left at all. Time had taken its toll. Those remains had decayed completely, and there was nothing left in those graves, largely because they were below the water table. Water had come into those graves, and water accelerates the degradation process. But there was some bone remains in one of the graves, and they were able to use that to extract DNA and make a positive identification after a false start, because they had misidentified it first, and then years later they repeated the test and identified the right person. So there were 3 misidentifications in this particular grave. They thought it was somebody, they did the initial DNA, thought it was somebody else, they did the final DNA, and they made a final identification at that point in time. And that just illustrates that this science of using DNA to identify people is evolving all the time. So the message is that DNA can remain intact for a long time, or it can decay very rapidly, depending on the environmental conditions. So cool, dry conditions are the best. So if you find remains in a dark cave at the very back of a cave, that's excellent. You know, that kind of environment will protect the remains. The remains at Tuam, a lot of them may be gone already. And the reason for that is that the pit, which was a sewage pit, was still in use up until about 1937. After that, it stopped being used as a sewage pit, but I don't know whether water was still going through that sewage pit or whether there were other environmental conditions that caused the remains to decay. So we don't actually know what we're going to find until we go and try and extract DNA from these remains. Also, just to make the point, this is a very well preserved hand from one of the bog bodies. Now, we find these bog bodies in bogs all over Ireland from time to time. They are thousands of years old and they have no DNA because the acidic conditions in the bogs destroy the DNA. So it very much depends on what those conditions are on the Tuam site, whether or not we can extract any DNA from those remains. So that's DNA extraction. Moving on to DNA testing, there are three main types of DNA. We saw this morning: Y-DNA, mitochondrial DNA, and autosomal DNA. And they're good for both deep ancestry and recent ancestry. This is what a chromosome looks like. So you see the classical double helical structure of the chromosome, and then the various bases, guanine, cytosine, adenine, and thymine, or what the letters stand for. G always binds with C, A always binds with T. And there are two types of Y-DNA marker. The first is a string of letters, TAC, 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 repeated 3 times. The other one is a single nucleotide polymorphism, where it's just a single letter that is affected. So think of one of the types of markers as a string of letters, STR, and the other one as a single letter. So those are the two different types of DNA marker. What are the options for degraded DNA? Well, you can use standard forensic tests such as Y-Filer 17 or PowerFlex 23 for the Y chromosome. They measure respectively 17 markers and 23 markers on the Y chromosome. For mitochondrial DNA, there's HVR1, HVR2, and the full mitochondrial sequence, which is exactly the same as what we do in genetic genealogy. For the autosomal DNA, there's a 17-panel identifiler test, and that focuses on STR markers on the autosomal DNA. Then you can use what we use in the commercial direct-to-consumer type tests. Y-DNA, we have 111 STR marker tests. We also have SNP tests which test up to 85,000 SNP markers. Look at the difference between the number of markers we test for our genealogy and the number of markers the forensic geneticist tests for crime scene investigations. They've got 17 markers, we've got 111. They've got 23 markers, we've got 85,000. There's a huge difference between these different approaches. Going on to mitochondrial DNA, we're exactly the same as the forensic geneticists. And going on to autosomal DNA, they use 17 markers, we use 700,000 markers. So there's a huge difference in these two types of approach. And it's very important to get the approach right if you're going to get the right results. Ancient DNA uses whole genome sequencing, and we're not there yet on the genetic genealogy side. We can do it for the Y chromosome with the big Y test, but we're not doing it for all of the chromosomes at this point in time. But it is coming, and we will eventually be able to have lots of people doing the whole genome sequencing, not just looking at 700,000 markers, looking at 3 billion markers across your entire genome. There have been major advances in this regard in the ancient DNA field, and those advances are informing what we do in the genetic genealogy field and also informing what we do in the forensic genetic field. So these rapid changes are really making us change the way we think. In terms of Tuam, I think the best options are probably going to be a chip-based technology like the 1.2 million SNPs that is being developed by David Reich's group. And then whole genome sequencing is the other option, using the same kind of techniques that Professor Dan Bradley uses in Trinity College Dublin. and the same techniques that Jans Carlson used with Thomas Kent in University College Dublin. So, like they said in their letter to the newspapers, we have the technology within Ireland. It's just a question of using it. And fast. So that's the DNA testing. Now, once you have extracted the DNA from the remains, the next thing is to compare it to something else. Ante-mortem samples. You might have heard of the case of Princess Anastasia, who claimed to be the last surviving child of the Bolsheviks in 1917. They were allegedly killed, and then suddenly 20 years later she emerged and she claimed to be Princess Anastasia, and it hit the headlines, and half of the nobility said she's definitely the princess, and the other half said no, she's an imposter. Well, she lived out her life with about 70 cats surrounding her, and married a historian, and they lived a very secluded life. And after she died in 1984, they dug up the Romanov, they found the grave of the Romanovs in 1991, dug them up and did some DNA testing. But the question was Princess Anastasia related to the Romanovs they found in the grave? What they did was, she'd had bowel surgery in 1979. There was still a segment of bowel preserved in the hospital. They got that, they extracted DNA from it, they compared it to the DNA they got from the Romanov site, and they found out that she wasn't in any way related to the Romanovs at all. She was an imposter, and her real name was Anna Anderson, and she had disappeared from Berlin in 1917 and had tried to commit suicide. She had her fiancé in World War I was blown up in front of her. So she's very, very traumatic background. And so that's an example of how an ante-mortem sample was used to refute the identity of Princess Anastasia. For the Fromelles situation, the 250 soldiers in the mass grave, they knew that they had to be on a list of 1,650 soldiers that were missing and that they had never been identified. But which of the 1,650 soldiers were these 250? And to answer that question, they researched the family trees of all 1,650 people. They built those family trees, and they built them down to living descendants to find 2 people who could give Y DNA, 2 people who could give mitochondrial DNA, and in that way they took 6,000 samples to identify 250 soldiers. So that was an example of how you target people for your comparison database. And the third way that we can look at data for comparison is you look at databases from the general population, and they may be forensic databases like the CODIS database in America, and there's various countries around the world, you'll see them in red there on the map, which have national DNA databases which are related to crimes and forensics, and it's possible to compare the DNA to these databases. Also from a genealogy point of view, we have the GEDmatch database, which is a public database, and I'll be talking about that in a moment. So once you've got your DNA for comparison, the next step is to establish the identity of the people involved. Now with Richard III, we targeted 2 of his relatives. We built the family tree back from Richard all the way down to, on the direct female line, to Michael Ibsen and Wendy Duldig. And they had a very close match, or an exact match, to Richard III's mitochondrial DNA that was extracted from the bones in the car park. But using the mitochondrial DNA alone only gave a probability of 92% that these were the remains of Richard III. It was the combination of the DNA with the other evidence, the injury to the skull, the high mineral content of the bones, the position that he was buried in the church. All of this other evidence meant that eventually the probability was raised to 99.9994%, and that was by use of Bayesian probability statistics. Those of you who are mathematics will understand what I'm talking about. I don't understand what I'm talking about. So this just points to the fact that we do need to have the relevant expertise to analyze the data and interpret the results. When we turn to Fromell, the way that they did it, and here is, if you like, a soldier born in 1895, that's his family tree. They went out on either side, maybe looking for his brother's descendants to see if there was any nephews, nieces, or grandnephews who would actually provide a Y DNA. They did the same on the mitochondrial DNA side and looked for mitochondrial DNA donors. If they couldn't find it by going out to his brothers and sisters, they went back an extra generation, and if they couldn't find them on the first cousin level, they went back to the second cousin level. So this was a huge amount of work, and you can see that they even had to build the trees out to 1800 and to go back up to come back down. In the end, then, what they were able to put together was a combined probability measure. So the probability on the mitochondrial side multiplied by the probability on the Y side gives a much stronger probability measure than either alone. But of course, this method would not work in Tuam because we don't have the father's side, because the majority of the children in Tuam will be illegitimate. And so we'll only be able to do the mother's side of the family tree. And that reduces the power of this specific technique to actually identify the children at Tuam. And now we turn to Dan Bradley and the work that his team did at Trinity College Dublin with the Rathlin Island skeletons. And he used whole genome sequencing. And this was one of the first times that it was used on such ancient DNA. This is DNA from 4,000 to 6,000 years old. And he looked at all 3 billion letters in each of these sets of remains, and it caused a minor revolution and has generated a lot of additional research since then, as a result of which people like David Reich, who has an ancient DNA lab in Harvard, I think it is, he is developing a chip of just 1.2 million markers that may be as costly as $200 per sample. $200 is very, very cheap for this type of work. Of course, it depends on the heavy throughput of the samples, but it could be in the next few years that we see that the new technology, which is better than the old technology, may even turn out to be cheaper than the old technology. And that's an important point to consider as well. Jens Carlson has a lovely little video on YouTube. I'd encourage you to watch it, about the identification of Thomas Kent and how they compared the DNA that they extracted using whole genome sequencing with the DNA of Thomas Kent's nieces. And they too came to the conclusion that the probability that Thomas Kent was in fact in the grave that they found, the probability was 5 trillion to 1 better odds. And just for those of you who are forensic geneticists, or if there's anybody watching this on YouTube, because it is being recorded for YouTube, these are the results from Ballynahattie and Rathlin, and you can see that the final endogenous DNA content was between 38% and 67%. And these are in skeletons that are up to 6,000 years old. And the mean coverage, they did a lot of, they focused on the first 2 skeletons and got coverage of 10.5, which was very, very good. And then with Thomas Kent, 26% of the DNA recovered was human. It was a relatively low read depth, only about 1 read per SNP. There were virtually no heterozygotes, but they analyzed the genome as if it was a half genome and just considered it as homozygotes. And they, like I said, the odds that the identification was wrong was less than 1 in a million, and the likelihood ratio was 5 trillion to 1, which is more than the number of people who've ever lived on the planet. So they were very confident with those results. In the last 4 weeks, there have even been more advances, rapid advances in this whole field. And I want to talk briefly about the identification of Buckskin Girl. Now, her name is actually Marcia L. King, but for 37 years, she remained unidentified. There was a young woman, about 21 years old, who was killed, left on the side of the road in Ohio. And for 37 years, forensic scientists tried and failed to identify her. So about a year or 2 ago, she was taken on by the DNA Doe project, which is a project that has been set up by a couple of genetic genealogists, just like us here. And they were able to arrange for, they found a 37-year-old blood sample that had not been refrigerated since 1981, and they were able to organize for it to be sent to Full Genome Corporation, where they managed to extract DNA from the sample. And then they created multiple profiles. They ran it through the machine again and again and again, and created multiple genetic profiles of this Jane Doe. And as a result of which, the number of no calls ranged from 23% to 44%, but about 50 to 75% of the SNP markers that are used in the usual kits that we use, Family Tree DNA, Ancestry, 23andMe, they were preserved. And what they were able to do was extract those from the profiles and build a spoof kit. And this spoof kit was uploaded to GEDmatch. And all of her top matches were present in all of the profiles, because they did the spoof kit for every single profile generated. And a typical profile looked like the following. GEDmatch has a template for about over 900,000 SNP markers, and an average or typical kit from Buckskin Girl was 595,000 SNP markers. The heterozygosity index was about 19.5%, and the number of no calls was about 35%. So that's very similar to what we were getting with Ballynahati and Rathlin Island. It was uploaded to GEDmatch and compared to the 800,000 profiles that are on GEDmatch, and a possible first cousin, once removed, was identified. With that information, they went on to Ancestry and they found a tree that corresponded to the results on GEDmatch. And within a short period of time, one relative in that tree was described as missing, presumed dead. So after 37 years of forensic science not being able to identify this poor girl, within 4 hours of her DNA being uploaded to GEDmatch, a positive identification was made. And that's absolutely incredible. The power of combining DNA and genealogy to actually identify missing people who have been unidentified for 37 years, absolutely incredible. Now, the interesting thing is that her mother was traced and contacted. She had never changed her phone number because she always was expecting a phone call from her daughter. So the take-home messages are there are major recent advances in DNA technology. Autosomal DNA extraction is possible. Sufficient SNP profiles can be generated for comparison with samples from living people, either targeted relatives or the general population. And the approach is similar to that done by the commercial direct-to-consumer companies and GEDmatch. DNA from a prospective family can be compared against the entire database of remains in a mass grave, and a list of matches generated so to enable identification. So it's a process that we are using today, and we can use it at the Tuam site as well. There are ethical issues around identification. So, for example, before qualifying for DNA comparison, people must have reasonable grounds to be related to one of the children at Tuam. But what are reasonable grounds? Where do we draw the line? First cousin? Second cousin? Who knows? Who decides? Who qualifies? Is there an ethics committee? Is there some kind of multidisciplinary board that decides you can compare your DNA but you can't? And what if some family members want to test and others don't? How do you resolve those conflicting desires? Another very important question is there a case for identifying all the children and not just selected children? Do all the children deserve to have their name on their gravestone? I would think they do. Do their families deserve to have their privacy protected? I would think they do. Is there a potential conflict between these two rights? Yes, potentially there is. So how do we minimize the risk? How do we minimize the harm that might result from this kind of conflict? This is the sort of thing that we need expert advice to help guide the discussion. Could the identification be done without publicity? I think that's very doable. I think it's very doable indeed. So, for example, if we were to use GEDmatch to identify these children, all we'd need to do is, first of all, extract the DNA from the children at Tuam, those that could give us DNA. A script could extract the DNA markers used on GEDmatch. That file would then be uploaded to GEDmatch in the usual way. But would this be the entire GEDmatch database, or would we have an opt-in version of the GEDmatch database for this particular project? Matches could be generated for each child. Some will have trees on GEDmatch, some will have trees on Ancestry, and we can move across to the Ancestry database and look at the trees there, MyHeritage, Genie, WikiTree, all the other ones. And we could even contact people for genealogical information, but all done in a very sensitive, diplomatic way. The identity of each child could be deduced, and then just like we do with adoptees, we can then approach close family and say, could you do a confirmatory DNA test to confirm that this child did belong to your family? And we've done the same technique with adoptees for many, many years. So just run through these very quickly. This is what GEDmatch looks like for those of you who haven't seen it. These are my dad's results. I have, even though it's a public database, I have privatized the names here and the email addresses. We also have the shared Centimorgan project based on 25,000 known relationships, so we know the range of centimorgan values for a given relationship. Here you're looking at half-siblings and first cousins. And this is the range of centimorgan values you get for these relationships. And we have 1,783 half-siblings, 874 first cousins who have donated their data for this analysis to take place. It's been made into a tool where you can actually plug in the number of centimorgans, and it generates the probability that it is a particular relationship. And we have that for parents, we have it for siblings, we have it for nephews and nieces, we have it for first cousins, and we have it for second cousins. And you see when we get out to second cousins, the probability of other relationships is possible because you're looking at smaller and smaller amounts of DNA. But the take-home message is probabilities for each relationship can be calculated statistically. And it's the statistics that is going to be a very important part of this identification process. The last ethical issue is memorialization. How do we create a memorial? What happens to the remains after we've extracted the DNA, we've done the DNA testing, there's been a post-mortem on the remains, there's been an inquest by the coroner. Are they buried? Are they put in storage? What do we do with the remains? And if we are going to bury them, what sort of graveyard are we thinking of? Are we thinking of gravestones that have a DNA identification number and no name on the gravestone? Or do we put the name on the gravestone and at a later stage, if they've been identified, we place the remains in that particular grave? These are all questions that we don't know the answers to at the moment. This is the sort of thing that they did at Fromelles. And you know those World War I cemeteries, they're laid out with beautiful military regimental order. And so far, 159 of those 250 soldiers have been identified. And every year they identify a few more soldiers. So there's a little bit of a ceremony where they take one of the gravestones that just has written on it, "A soldier known unto God," and they replace it with a gravestone with that particular individual's name. So we could do something similar like that at Tuam, and in so doing we give the child a dignity in death that it may not have had in life. Thank you very much. Just want to acknowledge a few people. John Cleary, who is sitting here in the audience, has done a lot of work on ancient DNA and is writing our ISOG Wikipedia page on ancient DNA, a fascinating read. And Catherine Corliss, that I've spoken to on the phone twice, Debbie Kennett, another one of our colleagues, Martin Curley, of course, who set up this whole conference, and I've had wonderful conversation with Martin as well about possibilities at Tuam. Dan Bradley and Jens Carlson from the Ancient DNA Labs, and of course the expert technical group who wrote the report, and also my ISOG colleagues on the various Facebook discussion groups who've been a source of great information and advice. So thank you very much to them, and I'll just leave that last slide up in case there are any questions.
I'm looking at the time here, so I think we'll have some questions for Morris if there's anyone else has questions. Then after we have the questions about this specific topic. Then I'll invite Jared and Seamus and Paddy to come up as well, just for an end of day question and answer session for everyone. If I did promise 3:30 into the conference, I don't want to tie people down too much. We've got to push through it as well. We've got to move about and get ourselves sorted. So, some questions for Morris.
Michael had a question over here.
Yeah, I suppose, having represented the Tuam area in the County Council for 15 years, and I'm now councillor in the Bannistow area, I did, in the course of my time in politics, have a lot of, quite a number of people who contacted me for their relatives, sisters, brothers, and had the opportunity, if you like, to bring some people to meet their brothers and half-brothers and all the rest of it, other people who were in a search mode. And I happened to be here at the council when the story about the commission report broke in terms of what happened to humans. It's a very morbid time in our history. But I suppose one of the things that I have been very careful about is in terms of that no matter what anybody says or what anybody does, the pain that a lot of those people that were involved in the people, the former residents of their home in particular, I don't think anything will take away the pain and trauma that they have suffered down through the years. I spoke to some of the people that you mentioned at length, and some of the people that you didn't mention that were involved in this campaign. How doable, and this is one of the things, and you've really tried to answer as best you can, how doable is it to identify almost every child, or indeed some adults that were buried at that site? And based on what you said, a lot of it is predicated on in terms of how much moisture or how much water was on the site or the location where the bodies were placed. Yes,
absolutely. A lot depends on what condition the remains are in. The short answer is we won't know until we actually try to extract DNA from whatever bones are there and see how successful it is. Now, we don't know to what extent the bones are scattered, but even if the bones were scattered, if the skull of the remains survived, then in the best case scenario, supposing there's nobody buried in that pit apart from the 796 children, we might recover 796 skulls, and then we'd use this new technique developed by the University College Dublin team to extract DNA from the petrous bone, and you saw Kendra Sirac extracting it there in one of the photographs. That has the best yield of human DNA, so the hope would be that we'd be able to get DNA from each of those 796 skulls, and that DNA those samples could generate a DNA profile that could then be used for comparison purposes. But we won't actually know the answer to that question until people go down into that pit, excavate it, and then find out what condition those bones are in. So until we've actually done the excavation, and this is why the expert technical group wanted to do the pilot study, because the pilot study would give them an indication of what to expect for the rest of the remains down there. They went in and took a small a bunch of remains, maybe 20 remains, and analysed that. If they got DNA from all 20 of those sets of remains, that's very encouraging. If they got DNA from none of the remains, that's very discouraging. But it gives an idea of how successful the process might be if they'd been able to do this pilot study before producing the report. And that, of course, would also help manage public expectations, which probably are very, very high, because people are watching television and they have CSI Miami and all of these programs where you get a little bit of DNA and suddenly the whole episode's over in 30 seconds because they've made the identification. It's not that simple. And a lot of people think it is. But I think you'll appreciate from the processes I've outlined here today that it is a very involved, very time-consuming, very complex procedure to actually get DNA from these remains in any mass grave. But because modern technology has advanced to the stage that it's at now, it is eminently doable. And the question is now, should we try?
on a point, what kind of costs would you be talking about?
To give you an idea, well, there were some costs outlined in the Expert Technical Group's report, and to do a complete excavation of the site, I think, was in the region of about 1.2 million. So it's actually quite doable from the point of view of excavation. From the point of view of actually doing the DNA testing, and that very much depends on what they're going to do. If they were to extract DNA and compare it against GEDmatch, for example, that would be a very cheap method because the database is already there. They don't have to do what they did at Fromell, which cost a lot of money, which was to track down 6,000 people to identify 250. And if we did have to do a kind of a targeted population that would form the reference database, then that could be a very, very expensive way forward. So I think the costs in terms of the DNA will vary hugely depending on what decisions are made. If reunification is a very, very important thing and they want to bring the bones of each individual back together, they may have to test every single individual bones, and that would skyrocket the cost of DNA testing. So that's why where do you draw the line is a very important question, and we don't know the answer to it yet. Now there was a question here as well.
You brought up the Romanovs, which brought me way back. This is going to sound crazy. The guy who was the head of that expedition was this Captain Peter Sarandanaki. I used to work with him. I was a ship captain myself. And just shows you how far this DNA's come. This is in the mid-90s. I had to drive with him. We went to a class in Baltimore. I had to drive him all the way back to New Hampshire. It's like 8 hours. And this crazy story about how he's going to find the Tsar's bones. Because his great-grandfather was a famous, I'm looking at it right here. If you guys want to look this up, I just found this. He had this website called Search, about searching for the Romanov children. And sure enough, he found them. He said this was his life's goal. His great-grandfather was a Tsarist general. He escaped out of the country, and he went back, and it was on Smithsonian, and he was the head of it. And I didn't think this. I said, this guy's crazy. Just about 6, 8 hours, I had to listen to this, and this is how far we've come. 25 years.
I know what we can do now by combining DNA and genealogy is absolutely amazing. You know, and it just showed that the Marcia L. King case, the Buckskin Girl case, which only broke 4 weeks ago, the fact that it defeated modern science for 37 years, and within 4 hours genetic genealogy had the answer, that's incredible.
Well, they just got another guy in the States with DNA. Who was up in California?
Yeah.
That's right. The Golden State Killer.
So there's a lot of people that are pretty nervous right now.
If you're a criminal, you should be nervous, you know, because the likelihood is that if this becomes widespread, you know, it will actually incriminate people and put them behind bars, which is probably where they deserve to be. I think what's even more encouraging about this development is that guilty people who have been placed behind bars because of a miscarriage of justice, or because of a misapplication or misinterpretation of witness testimony, an eyewitness testimony, terribly unreliable, bite marks, terribly unreliable. But you get DNA coming in now, and they're actually being exonerated of their crimes. And some of them have served up to 39 years in prison for a crime they did not commit. And they're being released, you know, and the Innocence Project was set up specifically to address the fact that a lot of people in prison today, in the US as well as in other places, have actually been put behind bars for the wrong reasons, and they have been inaccurately convicted of a crime that they did not commit. And the Innocence Project uses modern DNA techniques, and so far they have helped liberate and exonerate 351 people, 20 of whom are on death row. So the ability of DNA technology in combination with public databases like GEDmatch to liberate people who've been convicted of crimes they didn't commit, there's a huge potential there to actually help liberate a lot of innocent people. And I think that's a more exciting aspect of this recent advance compared to putting people away. I mean, it'll also help, obviously, if there's a serial rapist or a serial killer out there. It will make society a safer place if these people can be identified quickly and put behind bars, so they stop harming other people. Paddy.
What caught my eye was your very first slide with the death certificate of the son of a farmer.
Yes.
A considerable experience of looking at civil records say that farmers were male in the 1930s. So this child had a known father, an acknowledged father, and probably was not illegitimate.
This is where Catherine Corliss will probably come in as the local historian as a very useful source, because I think a lot of the children in the Tuam home were actually, their upkeep was paid for by the family of the father.
Maybe that's the case. I've gone through a small sample from the county nursery in Kilrush, which was a similar institution, and out of 43 there, 9 seem to have had fathers acknowledged. So I wonder, have we the 796 birth certs to go with the 796 death certs, and tell us how many of these were illegitimate, how many were orphans, how many were in there purely because of economic circumstances, how many were there, as in Kilrush, because of domestic violence, and the children and their mother were taken into the home. It wasn't purely for illegitimate children, I suspect.
I think that is research that is eminently doable,
Yeah.
you know, and I think we can do it. But as with all ethical questions, just because we can do it, does it mean we should do it? You know, and that's one of the questions, because one of the things that I think a lot of people want to avoid is a culture of naming and shaming the families that put them in the home, because I don't think that serves anybody's purpose at all. I think it would be counterproductive to have that kind of attitude. So it is one of those potential for harm situations where we have to risk minimization. We have to have a risk minimization strategy where the risk of harm is reduced and hopefully neutralized so it doesn't actually happen.
Morris, thank you very much. Wonderful presentation. I guess in just wrapping up there, and if there's any questions for any of the speakers today, DNA does not lie. And the problem is, as Morris just said there, how do we deal with the truths that come out about that? And there are going to be matches that will pop up in our DNA tests that we're like, how are they related? Because that's not who is supposed to be there. And it's a lot of just letting that, and as you said, the ethics of it, and we're just at the beginnings of that. So do that. I'm very conscious of the time here. As I said, there's a number of people who have signed up for a bus tour, and there's a number of people that said, oh, 3:30, we're going to be finishing up at. So I'm always conscious of that. Is there any questions for any panelists today that anyone has with regard to anything? I'm going to just people who haven't maybe spoken yet, and to just kind of like have a bit of a say pass.